Hello everyone and welcome to this presentation describing the research projects conducted by the members of the Canadian HIV Cure enterprise. If you're watching this video, you are surely aware that a major fight against HIV has been going on for nearly 40 years by scientists and communities. Since 1981, HIV and AIDS have represented a real public health challenge. Despite the constant decline in new infections around the world, it's still impossible to eradicate the virus. Antiretroviral therapies, ART, have been a considerable turning point in the fight against HIV, but they unfortunately cannot be considered as a cure. Do you know why? We know that taking ART controls the replication of HIV and reduces the quantity of circulating virus to undetectable levels in the blood. At this stage, infected individuals no longer transmit the virus. However, once ART is stopped, the virus comes back at a high level. This means that even during treatment, HIV persists but is not visible. But then, where does HIV hide? The virus persists in different organs which are themselves made of different cells. The target cells that HIV can infect and which can become viral reservoirs for the virus are primarily CD40 lymphocytes, but also macrophages, dendritic cells, and astrocytes. CD40 lymphocytes are cells that control the immune response by interacting with all immune cells. The cells have the HIV-1 receptor CD4 in large quantities, as well as the CXCR4 and CCR5 co-receptors. They are a major reservoir of HIV-1. Macrophages are immune cells that can be infected by HIV-1. Their cells can eat infected CD4 T cells, and this is the root of HIV-1 infection. Dendritic cells can be infected by HIV-1 and are also able to interact with an infected immune cells to transmit the virus. So, how are these reservoirs maintained? First, it's possible that ART does not completely block viral replication, or that some cells still produce HIV even in people on treatment for years. This is called the active reservoir. On the other hand, some cells contain sleeping viruses that we call latent. These latently infected cells can proliferate to ensure their survival or to respond to microbes. Thus, both the active and the latent reservoirs contribute to the persistence of HIV during antiretroviral therapy. Several strategies have been proposed to target viral reservoirs under ART. Here are two of them. The first strategy is to block the projection of new viruses by the active reservoir and then to lock infected cells permanently to silence the virus forever. Another strategy is to shock latent reservoir cells to wake up their viruses, making them visible to the immune system that can now kill them. However, these strategies primarily target CD40 lymphocytes, but HIV persistence is more complex than that. As I told you, HIV interacts with several cell types. Some cells can directly contribute to viral persistence by being infected, but other cells can contribute indirectly by interacting with infected cells and subsequently promoting viral persistence. Therefore, finding a cure for HIV will only be possible by understanding infection as a whole system and untangling these complex interactions. The CanCURE program contributes to the development of a cure against HIV by addressing three things. First, we want to understand the dialogue between the different cells that promote the persistence of infected cells. We also want to identify the cells in which HIV persists and their location in different organs. Finally, we want to develop novel cure approaches based on these findings and to test the strategies in animal models and in clinical trials. The CanCURE program members are proud to present you with their research projects. And we will start with theme 2, in which we want to identify the cells and organs in which HIV persists.
Hi, I'm Caroline Dufour, a PhD candidate in Nicolas Chaumon's lab at the CFCHUM. I've been studying HIV persistence in deep tissues. This is of interest since deep tissue reservoirs are only accessible when people living with HIV give their bodies to science. The goal here is to define how much HIV persists in different parts of the human body, which is an important step to be able to specifically target the tissues in which most of the live virus is found. So the main question is, where does HIV hide during antiretroviral treatment? We were able to get a glimpse of the answer when two generous persons gave their bodies for research on HIV reservoirs in 2018, allowing us to collect organs during autopsies. We looked for HIV in a variety of lymph nodes in different parts of the gut, but also in the spleen, the liver, the lungs, the brain, and the testis. And we found traces of HIV everywhere we looked, meaning that HIV reservoir persists in various deep tissues. But even more interestingly, we have developed in our lab an approach that gives us HIV identities. HIV identity is like an ID card. Knowing the virus genome gives us a lot of information and allows us to distinguish them individually among a whole population. This is important since there are only few live intact viruses that persist during ART, but these are the only ones causing viral rebound. This way, we can determine if the different HIV viruses we detect in the deep tissue reservoir are intact, alive, or defective, dead. We observed that as shown previously in blood, most of the viral genomes in tissues are actually defective. Finally, since we have a large number of different HIV IDs, we ask, can reservoir cells circulate between organs? Many viruses were found only in one location. However, some groups of identical HIV viruses were found in many organs, suggesting that reservoir cells can travel throughout the body.
My name is Julien Klein. I'm a PhD student in Dr. Jérôme Estaquier's lab at the CHU de Québec. One of the major obstacles in the HIV field is trying to understand why antiretroviral therapy is not able to eradicate latent virus in tissue reservoirs. That's why my project aims to evaluate viral persistence in liver and lung in HIV-infected rhesus macaques, which we use as our study model. Because liver and lung represent viral anatomical targets during HIV infection, it's of crucial importance to know whether these deep tissues represent viral reservoirs leading to viral rebound after ART interruption. The main objective is to define the role of tissue-resident macrophages, called TRMs, as viral reservoirs in the liver and lung. Thus, we will be able to identify and to target tissues and cells in which the virus persists despite ART. In the same way as HIV, the similar virus in monkeys called HIV can infect different cell types in multiple organs, potentially representing viral reservoirs. The study of rhesus macaques allows us to analyze deep tissues that are difficult to access in humans. But why did I decide to focus on the liver and lungs? Liver and lungs are actually constituted of specific immune cells called tissue-resident macrophages, TRMs, which play a role in tissue homeostasis or steady equilibrium. And they are different from circulating monocyte-derived macrophages, which play a role in tissue injury and inflammation. Today, compared to monocyte-derived macrophages, little is known so far about tissue-resident macrophages and their contributing role in HIV infection. To achieve this, how do we proceed? First, the monkeys are infected with simian immunodeficiency virus HIV. Then, we treat them with early antiretroviral therapy for 8 weeks. Unfortunately, the rhesus macaques have to give up their lives to recover the cells from the liver and lungs. Then, we isolate the tissue-resident macrophages from liver and lung tissues with a machine called cell sorter. Finally, by viral quantification, we are able to determine if tissue-resident macrophages are infected or not, and if they represent viral reservoirs.
Hi everyone, I'm Naz. I work in Dr. Para's lab where we study the impact of antiretroviral therapy on brain HIV infection. We are located at the University of Alberta in Edmonton within 3D6 territory. So far we learned that HIV hides in deep tissue. One of these HIV hideout places is the brain. It is important to study HIV in the brain because some patients experience movement and behavior disorders despite receiving antiretroviral therapy. And our aim is to measure how much HIV exists in the brain during antiretroviral therapy. And in a nutshell, we found that there is active HIV in the brain, and antiretroviral therapy is very poor at suppressing HIV in the brain. Let's see how HIV gets into the brain. HIV enters the brain either on foot in a fluid that goes to the brain, or it takes a shuttle by hopping on these cells that can enter the brain. Inside the brain, HIV infects microglia, which is a brain security force. Another cell that HIV can infect in the brain are these star-shaped cells that are important for the maintenance of the brain. It is interesting to know that HIV doesn't infect the nerve cells on its own, but the impact of infection of microglia and this other cell can damage the nerve and cause neurological disorders. Let's look at the consequences of HIV infection in the brain. As I mentioned, some patients experience HIV-associated neurological disorders. The rate of patients that experience these disorders was 1 in 2 before antiretroviral therapy, and after antiretroviral therapy, it is 1 in 5 patients. Our question is why the rate is not 0 after antiretroviral therapy? Is it because there is still HIV in the brain even after therapy? And how effective is the therapy to get rid of the brain virus? So to answer these questions, we looked at the brains of HIV positive patients, and also we looked at brains of infected monkeys, and we measured the virus in the brain before antiretroviral therapy, and we compared it to the amount of virus in the brain after antiretroviral therapy. And we found that these two amounts were similar, and interestingly, the drugs were not as effective in the brain. Which is quite opposite what we see in the blood, because the drugs are really good at suppressing the infection in the blood. So next, we looked at the impact of drugs in suppressing HIV in blood-specific cell type, the CD4 T cells, and the brain-specific cell type, the microglia. And we found that drugs cleared and suppressed the infection better in blood CD4 T cells compared to brain cells. And simply the drugs worked better in blood compared to the brain. So these findings can help with HIV cure, because if we know why HIV remains in the brain so stubbornly and doesn't get cleared and suppressed by antiretroviral therapy, it can help us to come up with solution. We can interrogate antiretroviral therapy, specific brain regions that are impacted, the immune response in the brain, and the specific cell type that can get infected in the brain, and we can design better drugs that are more effective in suppressing HIV infection in the brain.
Hello, my name is Jonathan Diaz and I'm a PhD student in Dr. Petronella Kuta's laboratory, and my work is focused on documenting the presence of long-lived and short-lived macrophages, a type of white blood cell in the immune system that fights infections in tissues of HIV-infected humanized mice models. My work explores their contribution to viral reservoir persistence during antiretroviral therapy. So our studies in humanized mice models demonstrate the direct contribution of myeloid cells to HIV rebound on antiretroviral therapy. However, the development of long-lived and short-lived macrophages in such models and their specific contribution to viral reservoir persistence during antiretroviral therapy remains unknown. Therefore, we believe that long-lived macrophages can harbor viral reservoir during antiretroviral therapy, while short-lived macrophages contribute to the primary HIV infection and viral rebound when antiretroviral therapy is interrupted. Therefore, if long-lived macrophages are identified as important viral reservoirs, then cure strategies should consider these cells as well, in addition to CD4 positive T cells. Interfering with HIV persistence in these cells may require specific interventions. So macrophages in the heart, pancreas, intestine, and skin are derived from circulating bone marrow monocytes and represent short-lived macrophages. While macrophages in the brain, liver, lungs, and epidermis are derived from embryonic or fetal precursors during embryogenesis or development of the embryo and from monocytes after birth and represent long-lived macrophages. So short-lived macrophages have a lifespan of a few days, while long-lived macrophages have a lifespan of a few years respectively. It is important for cure strategies to target these long-lived macrophages, as they have a capacity to self-renew and, along with CD4 positive T cells, can harbor viral reservoir despite being exposed to antiretroviral therapy for many years.
Hello, my name is Robert Lodge, speaking for Lee Jun Kong, and we work in the laboratory of Dr. Cohen. Lee Jun's project is entitled HIV VPU promotes infection of macrophages via increased engulfment. After my colleagues' presentations, I think that everyone now knows what macrophages are. My project focuses on the role of macrophages in HIV infection, and it is important because macrophages contribute to the development of disease in HIV-positive people by forming long-lived viral reservoirs. The main objective of this project is to investigate whether the HIV protein VPU promotes the taking up or engulfment of infected CD4 positive lymphocytes by macrophages through the reduction of CD47. In general, a greater understanding of macrophage infection by HIV is important for developing novel cure strategies. The level of CD47 on lymphocytes is a key factor in controlling their engulfment. When CD47 levels are high, it interacts with another molecule called SIRP-alpha, which is mainly found on macrophages. This interaction blocks the engulfment of the cells by macrophages. Our lab previously found that CD47 is decreased by the HIV protein VPU in infected CD4 positive lymphocytes. We have now subsequently proved that reduction of CD47 by VPU leads to more engulfment of these cells by macrophages. This is consistent with the fact that the level of CD47 in cells is the key switch that controls their engulfment by macrophages. Finally, and importantly, we found that increased engulfment of infected lymphocytes leads to greater HIV infection of macrophages and promotes better spread of the virus to other target lymphocytes.
Hello, my name is Oluwaseun Elizabeth, and I am a master's student in Dr. Thomas Maruka's lab. To address the theme of understanding the dialogue between cells that promote the persistence of infected cells, we have looked into how dendritic cells or DC synergizes with the signaling protein or cytokine called IL-7 to maintain latent infection in CD4 T cells. This is important as it would allow us to further understand the cellular mechanism in HIV latency establishment. And my objective is understanding the mechanism behind HIV persistence in HIV reservoirs. And of optimal significance, which is the implication my research would have for HIV cure, is that the understanding of the cellular mechanism which aids latency establishment may potentially lead to the development of improved therapeutics that may disturb disease or dendritic cells and IL-7 interaction with latently infected cells in order to prevent the establishment of viral latency in HIV-infected individuals. It is known that the main challenge in the development of an HIV cure is the presence of the HIV reservoirs, which are cells, mainly CD4 T cells containing integrated HIV genes, and they do not express any viral expression, and these are called latently infected cells. Also, HIV reservoirs could be referred to as the various tissues that houses these latently infected cells. One of the major HIV reservoirs is the lymph node. Here, I am depicting the lymph node as a photocopier that contains these latently infected cells. And in the presence of art, new viral copies are stopped from being produced, allowing for the management of the infection or disease. But when we stop art in infected individuals, this viral reservoir starts to make new viral copies from these latently infected cells. Thus, the persistence of HIV reservoir or latent cells hinders the complete cure or eradication of HIV. The microenvironment of the lymph node contains CD4 T cells here in blue, dendritic cells here in purple, which is another immune cell that is an antigen or disease-fighting cells. And it also contains high level of signaling protein or cytokine called IL-7. The microenvironment makeup helps to maintain CD4 T cells in the body, allowing for the normal functioning of the immune cells to help fight against infection and disease. But in the case where we have HIV infection, this normal physiological mechanism can be adjuncted by this latently infected CD4 T cells. And in fact, these IL-7 signaling CD4 T cells are individually known to help maintain their population through slow division without viral reactivation, so they do not trigger an immune response. So we look to see if these two signals work together or in synergy to help maintain the population of latent cells. And we saw that yes, there is actually some interaction that allows for the expansion of this latent population. And we are currently looking into the mechanism by which they do so. Therefore, it can be said that dendritic cells synergizes with cytokine IL-7 to maintain latently infected CD4 T cells. Thank you.
Hello, my name is Armando Espinoza Ortiz, and I'm a PhD student at Nicolas Chamont's lab. So my work is focused on the intrinsic relationship between the immune system and the HIV reservoir. In particular, I'm interested in the role of the third party, that is the whole community of microorganisms that live within and with us. So this project is aimed at a better identification of the HIV reservoir. Indeed, we know that despite the efficiency of ART, HIV persists in a latent form in certain cells. When the virus is in this latent or sleeping mode, to find which cell is infected is an elusive goal. Characterizing those cells carrying sleeping virus would be a major breakthrough. One lead to that goal would be through the antigen specificity of the infected cell. So, what is antigen specificity? HIV infects a very peculiar type of cells called T cells. They are one of the jail guards of the immune system. They patrol throughout our body, and thanks to the receptors, they're able to see pathogen or foreign invaders. Each T cell is specific to a very specific part of the pathogen that we call antigen. The issue is that when a T cell encounters an antigen, it will be activated and will proliferate. If the T cell is infected, the HIV reservoir will then expand. One of the main questions that our lab had is, are there specific antigens that are found in most people living with HIV? So we thought about what sources of antigen people living with HIV share. One of them is the microbiota. That is what we call all of the microorganisms that we live with. Those friendly bacteria help us in many ways. They help us maintain many natural mechanisms like digestion or strong immune systems. During HIV infections, there can be leakage of those good bacteria into the bloodstream. This phenomenon is called leaky gut. It is usually non-directly noticed by people living with HIV, and there are ongoing investigations to fully understand the mechanism and consequences of that phenomenon.
My work is also focused on identifying components of the microbiota that triggers the production of vitamin A on HIV replication in monocyte-derived dendritic cells. Our work has demonstrated that vitamin A produced in the gut in response to microbiota components can facilitate HIV replication and reactivate HIV reservoirs in CD4 positive T cells. By identifying bacterial components that trigger vitamin A production, we can help control the composition of the intestinal microbiota that can help reduce the size of the HIV reservoirs in people living with HIV. Dendritic cells can collect samples from bacteria found in the intestinal tract and can activate internal receptors. This will allow for the production of vitamin A, which will be directed to T cells by transfer proteins, allowing them to be more permissive to HIV infection. HIV could then integrate a genetic code in the DNA of the T cells and lead the T cell to infect other immune cells. Work also focuses on studying the effect of vitamin A on HIV replication in monocyte-derived macrophages. Monocytes are white blood cells that play an important role in the innate immune system. Our work has demonstrated that T cells carry high levels of integrated HIV DNA, while infection in monocyte-derived macrophages is rare in the colon of people living with HIV who are on antiretroviral therapy. By observing the effect on vitamin A on HIV replication, we can better understand the key roles in monocyte-derived macrophages during primary HIV infection and their contribution to viral rebound during antiretroviral therapy interruption. Vitamin A and its precursors obtained in the diet are absorbed by intestinal epithelial cells and can get converted into different forms and packaged into fat transport proteins. Fat transport proteins can then enter the general circulation and into the liver where they can store vitamin A. Vitamin A binds to these fat transport proteins in the liver and is carried through the bloodstream where it can interact with myeloid cells to allow the vitamin A to interact with receptors and regulate the activation of certain genes. In an environment where there is no vitamin A, macrophages are permissive to HIV infection. However, in an environment rich in vitamin A, like the colon, macrophages become a lot more permissive to infection and produce a lot more virus, and this can lead to the primary HIV infection and contribute to the viral rebound during antiretroviral therapy interruption in people living with HIV.
Hi, my name is Colin Graydon, and I work in the laboratory of Dr. Keith Folk. My research is focused on a braking mechanism of the immune system called LAG3, but I want to start by talking about HIV. HIV has 3 superpowers. Superpower number 1, immune dysfunction. HIV infects immune cells, throwing the immune system out of balance. And at the same time, HIV is a chronic infection, meaning that it sticks around, forever activating and forever exhausting your immune system. Superpower number 2 is mutation. HIV mutates a lot. This allows it to remain one step ahead of the immune system and makes it more difficult to design vaccines. Superpower number 3 is invisibility, aka latency. After it infects a cell, HIV can lay dormant, effectively hiding from the immune system. One strategy for a functional cure is known as shock and kill, which focuses on shocking HIV out of hiding and then killing the infected cell. We think that a molecule called LAG3 could help enhance both the shock and the kill components of this cure strategy. How? LAG3 inhibits cell activation, acting like the brake on a car. And it turns out that activating those latently infected cells can force HIV out of hiding. What's more, HIV-infected cells have more LAG3 on them than uninfected cells. This was discovered by the Chaumont lab as part of the CanCURE enterprise. So by blocking LAG3, we can disconnect the brake and potentially enhance our ability to force HIV out of hiding. That's the shock part. But the chronic activation caused by HIV infection exhausts the immune system. So even when HIV is visible, it's hard to kill the infected cells. But remember, LAG3 is a brake. And by acting like a brake on the killer immune cells, LAG3 is actually contributing to this exhaustion. So by blocking LAG3, we can disconnect the brake and restore function to these killer immune cells that can now kill the no longer invisible infected cells. In this way, blocking LAG3 can in part neutralize the immune dysfunction and the invisibility superpowers of HIV and enhance the efficacy of a functional cure. But LAG3 is still partly a mystery. For one, we know that LAG3 inhibits activation, but we don't know how. Secondly, LAG3 is a cousin of CD4, the HIV receptor, and it shares a similar structure. This raises the potential for LAG3 to maybe act as a sort of HIV receptor, enhancing HIV infection. To answer these questions, we created cells that do or don't express LAG3. By comparing how these cells differ during activation or how easily these cells are infected, we can answer these and other questions about LAG3 that are important to our understanding and may get us one step closer to blocking LAG3 as part of a functional cure.
Hi, I'm Augustine Fer, PhD student in Dr. Petronakuta's lab, and in our lab we are trying to determine if metformin treatment together with antiretroviral therapy could contribute to HIV remission or even cure. So even under antiretroviral therapy, people living with HIV are at greater risk of developing complications due to chronic inflammation. These health problems are mainly due to the constant activation of the immune system by HIV persistence. Antiretroviral therapy blocks viral entry and exit but does not block HIV transcription. This causes chronic inflammation and non-AIDS comorbidities such as diabetes, cardiovascular disease, or even cancer. New therapies are needed. Our objective is to determine if metformin, a common drug used to treat type 2 diabetes, may be used to reduce HIV-related inflammation and therefore limit the risk of developing non-AIDS comorbidities. Here is a replication cycle of HIV. So first the virus binds to the cells and it fuses with the membrane. Its genome is transformed into DNA, so from RNA into DNA, and then integrates the whole cell's DNA. Then HIV DNA is transcribed and translated in order to form a new virus. Next, the virus undergoes maturation to form a virus with the ability to infect other cells. A lot of these steps are blocked by antiretroviral drugs, as you can see here in blue. However, antiretroviral therapy does not block HIV transcription and translation. So what is transcription? It's a process to produce protein from DNA thanks to RNA. So proteins are molecules necessary for the proper functioning of the cells in our body. So the human DNA, also called genome, contains thousands of small entities called genes, and each contains the information necessary to build specific proteins. So how do cells fabricate protein from DNA? Thanks to RNA. So it's a smaller code that contains only the information of one gene. And RNA behaves like a messenger between DNA and protein. This is why it's called messenger RNA. To simplify, you can see it as a construction work. So the architect creates plans of the whole city, exactly as DNA contains all the information to produce all the protein of our organism. The architect gives the plans to the foreman, and this step is called transcription. And the foreman explains to the worker the plan of one building, just like RNA contains the code of only one gene. Then the worker builds the building in the same way that the ribosome will build the protein. This step is called translation. So what about HIV? The HIV genome, by integrating the host cell's DNA, behaves like a human gene. It will undergo transcription and then translation in order to form the new virus. So HIV transcription is not blocked by antiretroviral therapy, so it occurs during treatment. And the accumulation of HIV RNA into the cells causes chronic inflammation and immune activation. This process is called residual transcription. Our lab, in collaboration with CanCURE labs, has performed a clinical trial named LILAC study. And in this LILAC study, 13 participants were treated with metformin over a period of 12 weeks, together with antiretroviral therapy. And we have shown that 12 weeks of metformin treatment helped to decrease HIV transcription in 8 out of 13 participants. With antiretroviral therapy only, there is residual transcription, and this leads to inflammation. But by adding metformin, there is less residual transcription. This means that there is less inflammation. So this could mean that metformin can limit the risk of developing non-AIDS comorbidities. Thank you for your attention.
Hello, my name is Nicola Bellini. I am a PhD student in Dr. Eric Cohen lab, and I study the role of micro-RNA in HIV-1 infection. More precisely, my project mainly focuses on the CCR5 co-receptor of HIV-1. The level of expression of CCR5 has a central role in the susceptibility of CD4 T lymphocyte to HIV-1 infection and HIV-1 latency. Indeed, the only cases of cures of HIV-1 reported are patients who have a lack of CCR5 co-receptor on the cell surface and are therefore resistant to infection by HIV-1. However, the strategies used to achieve this are not applicable on a large scale. In this context, we are investigating how micro-RNA, a small molecule generated by the cell and that regulates cellular process, impacts CCR5 expression and HIV-1 infection. In fact, using micro-RNA, we propose a novel approach that renders CD4 T lymphocytes more resistant to HIV-1 infection, thus countering the establishment of viral reservoir. The level of CCR5 is variable on the surface on CD4 T lymphocyte and depends on the level of activation. This difference in CCR5 expression affects the susceptibility of lymphocytes to HIV-1 infection and therefore the susceptibility to the establishment of latent infection. In our lab, we are trying to understand what are the mechanisms behind this modulation of CCR5, and we believe that micro-RNA play a role in this mechanism. As mentioned previously, micro-RNA are a small molecule generated by the cells and that regulate cellular process. Recently, our laboratory discovered a micro-RNA, micro-RNA 103, that reduces the level of CCR5 on the surface of cells. The objective of my project is to investigate the role of micro-RNA 103 in CD4 T lymphocyte and to determine which cellular process control it. Our results show by increasing the micro-RNA 103 in the cells, we see a decrease in the level of CCR5 on the surface of CD4 T lymphocyte, preventing HIV-1 infection and indirectly the establishment of latent infection. Overall, this finding brings new perspective to counter the establishment of HIV-1 reservoir.
Hi everyone, my name is Isa Munoz Arias. I am a research associate in the laboratory of Dr. Eric Cohen, and today I am going to be presenting to you about my project titled The Role of VPR in HIV-1 Latency. Why is this project important? The biggest block to an HIV cure is the HIV-latent reservoir. Because of these, we need to study the factors involved in establishing or maintaining HIV latency, so that these latently infected cells can be more specifically targeted by drugs with the shock and kill technique. The objective of the project is to study the role of VPR in establishment or maintenance of latency. The implications for a cure is that targeting silencing factors directly with drugs or by enhancing VPR expression may contribute to the shock and kill technique. What is VPR? VPR stands for viral protein R. It is a protein carried by the virus and also released by productively infected cells. VPR depletes more than 1,000 cellular proteins, so by doing this, it changes the whole protein content of the cell. Our hypothesis, or what we think is going on, is that VPR may be sensitizing CD4 T cells for productive infection by depleting factors involved in establishing or maintaining HIV latency. How is HIV latency maintained? Before we answer this question, let's talk about a few facts about DNA in general. The length of all DNA in the human body is 108 billion kilometers. That is 150,000 round trips from Earth to the moon. That is a lot of DNA, which means that it has to be packaged to fit inside our tiny cells. And all of these DNA is not always active, so certain genes have to be silenced so that they are not all making proteins at the same time all the time. The way that DNA is packaged is that it is twisted around protein complexes called histones. The twisting of the DNA around histones compacts the DNA so that it can fit into the nucleus of the cell. The combination of DNA and histone is known as nucleosomes. Silenced DNA is twisted more tightly around the histone and has other protein complexes attached, blocking away any chance of the DNA getting activated. When HIV DNA integrates into our DNA, it acts just like our DNA. There is no distinction. So in latently infected cells, it too gets twisted around the histones very tightly, and then silencing complexes come and lock it down so that the HIV DNA is inaccessible for viral production. In reactivated cells, HIV is not locked down by silencing complexes, so it is exposed and ready to produce virus. What we think is going on is that productively infected cells secrete VPR and produce virus carrying VPR. When enough VPR gain access into latently infected cells, some silencing factors are degraded by VPR. When VPR accumulates in the cell and degrades the protein complexes, the HIV DNA is exposed and ready to produce virus.
Hello everyone, my name is Jaspreet. I am a postdoctoral fellow in the laboratory of Dr. Eric Cohen. My research work is entitled SMAC mimetics as novel latency reversing agents. This study is important because the most effective latency reversing agents tested with promising outcomes in the laboratories increase inflammation in ART-suppressed people living with HIV and therefore are difficult to evaluate clinically. Thus, there is an increasing need for the development of novel therapeutic approaches that can eradicate viral reservoirs without generating inflammation. Here we demonstrate in humanized mice that mimetic of second mitochondria-derived activator of caspases or SMAC mimetics help in the eradication of viral reservoir via the shock and kill approach. They reactivate HIV in a clinically safe way, and these reactivated cells are killed via activation of mechanisms mediated by the immune system. This makes SMAC mimetics safe for clinical testing. SMAC are generated by the cells on receiving stress signals and are important to realize cell death in affected cells. Mimetic of SMAC were recently developed as a new class of targeted drugs for the treatment of solid tumor or hematological cancers. In the field of HIV, SMAC mimetics have shown to have three superpowers. First, they activate non-inflammatory pathways to reactivate HIV and hence are clinically safer to evaluate. Second, they degrade proteins that are inhibitors of cell death. And third, they induce cell death in reactivated as well as in latently infected cells, and hence they might reduce the viral reservoir. All of these superpowers make SMAC mimetics a perfect shock and kill approach in the field of HIV cure. In this project, we evaluated the effect of SMAC mimetics in virally suppressed humanized mice. We started by infecting humanized mice with green fluorescent protein-linked HIV. After the establishment of infection, these mice were treated daily with antiretroviral therapy, commonly known as ART. On achieving complete suppression, the mice were treated acutely with SMAC mimetics in the presence of ART for 48 hours to 2 weeks. At the end of the treatment, mice were euthanized and various organs were tested for HIV reactivation and reservoir reduction. We observed that HIV reactivation and viral reservoir reduction occurs at varying levels in all organs used in the study. Also, at antiretroviral therapy interruption, we observed that mice that were treated with SMAC mimetics had a virus rebound of less than 10 to 100 fold. Altogether, these results demonstrate that SMAC mimetics are effective latency reversing agents with the ability to preferentially kill latent or reactivated cells in a clinically safe manner. Therefore, these compounds should be tested in combination with ART in people living with HIV. Thank you.